Hello everyone, and welcome to the lecture on immunodeficiencies. So we're picking up a little bit from last time where we finished talking about traditional vaccines. I guess these two aren't totally connected, but we're going to talk a little bit about immunodeficiencies, kind of link that to some of the work we've been talking about with B T cell development. And then talk a little bit about HIV because it kind of demonstrates some really interesting ideas about the immune system and vaccine development. Okay, so here we go with immune deficiencies, and this slide should look familiar to you because we're looking at stem cells and then maturation or activation of B cells all the way here, B cells and T cells. So here are the two, so B cells on this side, T cells over here. And what this is, this chart is linked up to is all of these different diseases that and the stages of development that those diseases affect. And so these genetic immunodisorders or immunodeficiencies really block. They can be traced to genes or mutations that affect development of, say, a pre-B cell into a mature B cell, or a mature B cell into a memory B cell, mature B cell into a plasma cell. All these different things can be linked to different diseases. And one thing I want you to think about when you look at something like this, if the higher you are up here, really the more severe these genetic deficiencies are. So take a disease like SCID, which is severe combined immunodeficiency. That is linked to a single protein or a single gene, a deficiency, but it prevents these progenitors from differentiating into pre-D, B, and T cells. So you really, these are kids that end up dying very early because you essentially have no immune system. You get some part of the innate immune system, but you have no humoral response. You have no adaptive immune response. So the farther you up here, the more lineages you are going to be affecting, and therefore kind of the more severe you're going to be. It's not to say that these are not severe diseases, but the more different types you affect, the more severe or the more quickly that effect will be seen. So this is taking that same chart we just looked at, and now breaking it down to here's SCID right here. So SCID can be, there are several different versions of it, but they have these deficiencies, and you look and you see some of the signaling pathways that we've talked about: IL-2, JAK3, ZAP70, which is a critical in creating the docking sites for signaling from T cell receptors. It even has a RAG1, RAG2 deficiency, which means there's no T cell or T cell receptor or B cell receptor arrangement. So you can see why this is such a dramatically severe disease, and why it can be why it has to be diagnosed very early. And they're even developing because there are such strong genetic linkages. And so some of these genes, depending on, I don't believe this means if you have SCID you have every one of these. It means these have been linked to SCID, so these different mutations. And so some of these mutations, if you have, let's say, an ADA deficiency and your SCID, gene therapies become very promising therapies for this, because if you can replace a deficient gene with a proper gene, you essentially are cured if you hit the right stem cells. Now you have to target this to stem cell populations, which in the bone marrow is not incredibly hard to do. You can do that with things like autologous stem cell transfers. And that's one of the ways these therapies are being used. There's a great study, I should have had it linked, sorry about that, but if you look it up, there was a gene therapy, targeted gene replacement for, or actually not targeted gene replacement, a gene replacement for I think it was something like 10 kids that had SCID. and 7 of them were completely cured by the replacement of the single gene. But 3 of them ended up getting essentially leukemia, which is the overproliferation of certain B cells, and that was due to the insertion of their vector, their recombinant vector, targeted cells and inserted at a particular hotspot, which sat right next to an oncogene and then promoted the development of essentially cancer. So, I think some of those trials got put on hold until they can target the genes better, but it shows kind of the promise for this thing. So you can look at, I'm not going to go through all of these different things, but you can look at, the Bayer lymphocyte syndromes, what's not expressing MHC class II receptors. You can have, different things that are X-linked versus autosomal recessive. So that's what X-linked there versus autosomal recessive means. And so you can see kind of some of these defects, and I thought this was kind of a nice slide because it leads to this one, which is even more complicated, that illustrates, kind of, I hope it kind of brings home a message of, we've been talking about all these different things. So these CD40 ligands. So here's the RAG1, RAG2. So this is the SCID, one of the SCID mutations. Here's the Bayer lymphocyte mutation right here. It kind of hopefully drives home some of the things we've talked in these things like BTK and this tyrosine kinase. So there's a defect there. There's that deficiency in the JAK3 and the JAK-STAT signaling. You have cytokine receptor mutations here. It helps to bring home, again, this, some of the things we've been studying and kind of put them in perspective that, we've been talking about in development, but when these things go wrong, that's where we see these immune disorders, and these disorders are obviously very serious and put people in jeopardy. So now I want to switch gears and talk a little bit about HIV virus or AIDS. And as it's an interesting thing, it's obviously an important thing to talk about in the immunology class, because these viruses affect immune cells, and particularly T helper cells. And so here's our kind of cartoon of the HIV virus. It has these coat proteins on its cell surface, so it's an envelope surrounded in a membrane from the cell in which it buds. So here's a particular cell which is budding out new viruses right here. So these are viruses budding off of this T cell. It has these GP140, GP120, GP41, which are important for targeting. It actually has MHC receptors, which come from the host membrane. Inside, it's carrying some different proteins and RNA components. So it has its RNA genome, so it's single-stranded RNA, which you're seeing here. It has to bring with it its own reverse transcriptase, because it is going to have to make a copy of that RNA and turn it into DNA. It has integrase, which is important because that's how the DNA that they make from reverse transcriptase is going to be integrated into the host genome. And that carries with it protease, which is important for cleaving up some of the proteins, which you'll see in a minute. So that's the basic structure of it. What does it look like genomically? So genomically, you can look at the virus and you see these big, it kind of sometimes is just broken down into these big segments: gag, pole, and envelope. And these 3 regions are polyproteins. So these are transcripts that are made, and those transcripts encode for multiple proteins on the same peptide. So you have a protease that will come and cleave these things at precise sites and make the individual components of these. So the individual proteins must cleave from the larger peptide. That's why the protease is such a target for therapies. Many of you have probably heard of protease inhibitors and protease inhibitor cocktails for HIV virus, inhibiting HIV virus, and that's the reason it's used, is because you need to, it's part of the life cycle of the protein. That's how you cleave it. If you inhibit that, then you can't assemble the full virus. And here is a breakdown a little more specifically. So here are the GAG. Here's the polyprotein. So it's a 53 kilodalton precursor protein here, and it makes these nucleocapsid proteins. And so these are the 4 proteins that it makes. Okay, envelope makes these transmembrane proteins, so those envelope glycoproteins. So that's the GP 120, 141. These are the ones that will bind and identify CD4 on T helper cells. And then the pol, which basically stands for polymerase, will make that reverse transcriptase and the protease and the integrase. So this is kind of has this polyprotein. So here it's the kind of the core of the nucleocapsid. Here's the envelope, how it will target and bind to its target cells, and then these are the genes that are important for replicating and integrating its DNA, or from its RNA to RNA. And so then you have some other proteins that are also important for its life cycle, and has some kind of neph, which I like to call it nefarious, because that's one of the bad, one of the evil things it does, is it down-regulates the host MHC class 1 receptors and CD4 receptors. So it's, basically it doesn't want its CD4, it doesn't want its T helper cell to die. And so T helper cells will die if they are recognized and activated in the immune pathway. So if you have a CD4, recognize an MHC receptor, and so it will then be activated and then start going through apoptosis. However, viral cells just want the cells to kind of be living and kind of in stasis, not being activated, not dying. So it's wise to shut down CD4 and MHC class 1, so it's not recognized by your immune system as a viral infected cell. So it's not going to present some of these viral proteins on its cell surface. Okay. So again, here's another one. This VPU degrading CD4. Again, kind of an evil way of surviving in our immune system. So you can see this virus is really fine-tuned, like most viruses are. But these, most viruses are finely tuned to their host cells and how they're going to maintain their life. It's really not in the best interest of a virus, any virus, to kill its host cell. So its goal is to live as long as the cell will manage to live, be passed down to any daughter cells, that cell if it divides, and make as many of its progeny as it can. So let's look at its integration into the T helper cell life cycle. So here we have a bit of the cartoon showing how HIV infects a target cell. So you have that GP120 and CD4. So GP140 binds CD4, and that's what targets it specifically to T helper cell. Through this process here, you have essentially a receptor-mediated endocytosis, and that viral envelope membrane then fuses to the outer membrane, leaving the encapsulated or the capsid proteins surrounding that single-stranded RNA. That single-stranded RNA has to be copied into DNA, so you first start with a hybrid. You then make double-stranded DNA there, which now, so that was our reverse transcriptase right there that is carried along with the capsid. And then you have the double-stranded DNA, and now it uses that integrase right in there, and where it can be stably inserted into the host genome. So it only can survive, integrated in the host genome. It can't survive out in the cytosol or in the nucleus without being integrated into the host genome. One thing I wanted to add is it's not just the CD4 and that is required, that you're actually using some of those co-receptors that we talked about. So CXCR4 is important for this as well, endocytosis of the viral protein. Okay, so that's the life cycle of it in terms of integrating into the host genome. Now, particularly the most important in terms of its ability to escape the immune system are those things like downregulating CD4, downregulating MHC class 1 receptors. But what is also important is this reverse transcriptase is a error-prone enzyme, and it does not have editing functions. So mistakes that are made here in this to make this DNA-RNA hybrid are stuck, meaning they're not going to be repaired. And so that makes this virus it is able to mutate very quickly. And so the fact that it has this reverse transcriptase allows it to mutate quickly and develop antigens that are new antigens that change its profile so it's not recognized by your immune system. Now the next part of the life cycle is the production of a new virus. And so now once in the host genome, it becomes transcriptionally active, and it's transcribed and translated by the host cell machinery, obviously like all viruses. And so you have to do two things, which is make proteins. So those gag, pull, envelope proteins are the major ones here. They're precursors that the protease will then chop up into the smaller pieces. You have single-stranded RNA here, which needs to be packaged up with some of these proteins in the coat proteins, along with the reverse transcriptase and the integrase and the protease. And then it assembles in essentially what is a coated vesicle on the cell membrane and buds off of the cell. So now you have an envelope enclosed HIV virus that contains its genome, its proteins, and its targeting receptors, which are those GP120s. So that's really the life cycle from infection to production of it. Now, what was kind of the mystery, and some of you are not old enough to remember this, but when I was in grad school, in undergrad, it was really people couldn't figure out what was happening to T cells and why was it so difficult or there's essentially a latency for the HIV where it couldn't be detected, and many people thought it was essentially hiding somewhere, it was not being produced, or your immune system couldn't fight it. But what's been figured out since is that the key point here is that infected CD4 cells are attacked. But what happens is they're just kind of continually decreasing and decreasing and decreasing, and it can take a number of years before essentially the HIV essentially kind of wins the war, meaning the viral load will essentially be kind of stable for a long period of time. CD4s are being produced in mass quantities, and it's held in check for years, and then finally you kind of just hit this breaking point where the T cells become too low. Then you become immunocompromised, and that's the immunodeficiency part of the disorder, and your viral load goes way up. So that kind of understanding, helped people understand how to develop some of the treatments. And so the therapies, once we understood kind of the cell biology and what was going on, how the virus reproduced, what its life cycle was, it was then able to start developing distinct therapies based on targeting different aspects of the life cycle of the virus. So what you see with the virus is the virus can hit, there are different ways to hit it, and there are obviously a lot of kind of not easy targets, but obvious targets, which is these enzymes that are unique to the virus. We do not have reverse transcriptase. We don't use integrase. And we don't use these proteases, these specific proteases, to cleave these polyproteins. So these became very, very good fertile targets. and then obviously kind of blocking the docking sites. That became a very fertile part as well. Those could change, as I mentioned, the mutation rate based on the reverse transcriptase or lack of editing function made these kind of a harder target to work with. But these became kind of the targets. How do you hit, and how do you create therapies that will affect the virus and viral infected cells, but not affect healthy host cells. So here are kind of some of the particular therapies, and you can see some of these. There were just a whole. You can kind of catalog. These were ones that were reverse transcriptase inhibitors, and so a lot of these are looking at making nucleotide analogs that essentially stop or prevent reverse transcriptase from working very well. And again, since we don't have reverse transcriptase, it is a good therapy to use. However, you can see from a lot of the side effects, it's not perfect. It's not like your cells don't mind these things at all. Again, here are more reverse transcriptase inhibitors. And when you hear things talking about cocktails, that's one of the, because you have so many different types of therapeutic agents, you can work with a lot of different types of people with genetic backgrounds, which might have one might be more effective than another. Here are a class of the protease inhibitors here, and again, you have a nice array of them. Protease inhibitors, we do have proteases in our bodies, but again, you need a specific protease inhibitor to cleave these polyproteins, which we do not do. So again, it becomes something that is a very good target. It's one of the reasons, so having these targets, these proteases, reverse transcriptase, makes this a virus that we can develop a lot of these therapies with, and so now HIV is not cured. You're not cured. None of these treatments cure HIV. You have an HIV infection, but what it does is makes it essentially a disease you can live with. You can live a very long time with these protease inhibitors, with these reverse transcriptase inhibitors. Now, as I mentioned, cell fusion is a little harder to deal with, partially because CD4 is a receptor that if you're finding effectors or things that can prevent CD4 binding, that might affect other cells that need CD4 binding, and then the mutation rate of the GP120 can affect the effectiveness of that. So that's one reason I think you don't have as many of these types of therapeutics. Now, if we look at the effectiveness of some of these therapies, you can look at how dramatically, once these therapies kind of came online in the mid-90s, and you can see the treatment levels have gone just dramatically down into the number of deaths per 100,000 people, which is about the same as homicides right there, and it's less than suicides, heart disease, cancer, and unintentional injuries. I like this category here. I'm curious as to what all those would be. So one thing, the kind of final slide I wanted you to think about is, well, we've created some treatment that can allow people to live a long life for the most part with HIV. But one thing we haven't created, this is a virus like many other viruses. We have vaccine, unlike many other viruses, we have vaccines for those viruses that prevent the infection. And right now there's no such thing as an HIV vaccine. It's just treatment once you have the disease. And so the question is, why is this the case? There are some good reasons, but there are some good reasons, and that's what I wanted to kind of go through here as to why um, that we don't, we don't see these things. So the ones I really want to highlight is that vaccines, most of our vaccines and things like chickenpox, that kind of, those polio, those things are against viruses that do not change. Don't change a lot over time. As I mentioned, it mutates very rapidly due to that reverse transcriptase. So that reverse transcriptase, the error-prone nature of that, makes it difficult to develop a vaccine. Kind of like the flu virus. The flu virus changes very rapidly. It's the same reason you have to be vaccinated every year for the flu vaccine. A lot of, the most effective ones are these killed or live attenuated viruses. And HIV, the problem is that it doesn't have any antigenicity. And that was one of the things we mentioned when we're talking about traditional viruses, is anytime you're, the farther you get away from the pathogen, the less likely or the more likely it is that some of those antigens will be different or altered in a way that will not make your immune response as robust. So another thing is its infections. That most vaccinations are due to airborne or mucosal surfaces, so sneezing, that kind of thing, respiratory, gastrointestinal, and due to HIV is bloodborne and through sexual, a lot of times through sexual contact. And the final problem that I wanted to mention is that there's really there are no animal models for us to really study that, and we don't want to do human volunteers for studying HIV vaccines as well. So it's really something, I think this is what there's been great the understanding the molecular basis of the immune system and the molecular basis of the virus have allowed us to develop very effective treatments, but still the vaccine, something that prevents infection, is something that's still out there, and so that's something that people are still actively researching, and hopefully that can be something that can be solved in the future.